Now, again, earlier we discussed that we have two different types of cells, somatic, which divide through mitosis, and gametes, which divide through a process called meiosis. So let's take a look at meiosis. Okay, so during interphase in meiosis, we begin with two homologous chromosomes, and those are two chromosomes that are the same in length, in like centromere location, and like DNA orientation, but they're from two different parents. So, and then each set of these homologous chromosomes during interphase duplicate. So like we have two pink, two yellow, but like these match up. Okay. During prophase 1 of meiosis, just like prophase of mitosis, the nuclear envelope breaks down, the spindle fibers begin to extend. However, the homologous pairs, and I'm just drawing one, by the way. There's, of course, multiple. So the two sets of sister chromatids, they link up, and at certain spots, they'll meet and do this process called crossing over, where they will exchange different certain sections of their DNA for the corresponding ones of the others. Just like metaphase of mitosis, we got the spindle fibers fully extended, except, so we have our pair of homologs. Spindle fibers attached to both, from one pole, attached to both of the kinetochores for one set of one homolog and the corresponding for the other. And the homologs meet at the metaphase plate in the middle because the spindle fibers have extended, pushing them together. Metaphase 1 this is. Then during anaphase 2, or anaphase 1, I'm sorry, same deal happens as in mitosis with the spindle fibers, the non-kinetochore ones causing the cell to lengthen. However, instead of pulling the two, just one like chromosome apart, we pull apart a chromosome pair. And to show the crossing over, I'll draw it half pink, half yellow. So those begin to be pulled apart. Then we have telekinesis and cytokinesis. And again, just like telekinesis, telophase and cytokinesis 1. Just like in mitosis, nuclear envelope forms. We have the centrosome on each side. The spindles have broken down because they shortened and pulled these. And then we have our chromosome pairs in each side. And then during cytokinesis, this cleavage furrow forms, causing these cells to split into two. From here, we enter meiosis 2. So meiosis 2 separates the sister chromatids. And it consists of prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. So the same 6 steps as the last ones, except 2. So again, during prophase 2, I'm just drawing one cell, but remember, we have those 2 separate cells. So this is happening in 2 cells. So in prophase 2, again, same deal with these guys. Start to expand. We get that nuclear envelope breakdown. These guys condense back more because they decondense during cytokinesis and telophase. And then during metaphase 2, they meet up at the metaphase plane. Oh my gosh. And we have our spindle. This time, the spindle fibers from opposite poles attached to the one kinetochore. And then anaphase 2. However, it's important to note that these 2 sister chromatids, as you can see through the color, like I said, unlike mitosis, they're not genetically identical because of the crossing over that happened. So yeah. During anaphase 2, we extend that guy. Our non-kinetochore microtubules are pushing the cell so it elongates, but the kinetochore microtubules are going to pull apart the chromosome at its centromere by shortening. And then telophase 2 and cytokinesis, same thing. However, again, this is all happening times 2. So really, we get, I'll show it in this one. We got our nuclear envelope forming, our centrosomes, our cleavage furrow that forms during cytokinesis. We have all this unique DNA because of the crossing over that occurred. And each of these cells, our chromosomes are starting to decondense. Nucleolus pops back up too, just like in telekinesis and, oh my gosh, telophase. And cytokinesis and mitosis. And we get 4 unique gametes, each with a half set of DNA, rather than a complete set. That way, the diploid will be restored when fertilization occurs, and we'll have 1 set of chromosome from the dad, 1 from the mom. They'll combine, we'll get that nice little 46 chromosomes, because each of these cells will have 23, and that's a very, very condensed version of meiosis. Thank you for watching this video. Sorry it's so long. Bye.